Alright, welcome back. We began this journey in our last video by asking a question. What does it take to build a human? And we discussed the 20,000 different genes that led to the production of 100,000 plus different proteins, and these somehow work together to create unique cell types. Well, an integral part of this process is how individual cells communicate with other cells and with their surroundings, what we would call their microenvironment. So as we introduce this idea of cell communication, broadly speaking, we can break this down into direct and indirect, where direct is kind of like a handshake. This is direct cell-to-cell contact, where indirect really can be defined by how far I want my signal to travel. And roughly, we can classify these into three different types. Autocrine, which is a signal I don't want to go very far at all. This is going to be a signal that comes back on me. This is a signal for self. The next will be paracrine, and this is a signal for everyone in the neighborhood that can respond to it, but it's designed to be local. We're going to stay close by. And that's in contrast to endocrine, which is meant to go throughout the system, and as an organism gets big enough, this is going to be primarily through the flow of blood. As we think of many hormones, for example, secreted in the blood, and then they get through all of the system. So this sets up a nice, albeit very simplistic and broad view of how cells communicate. But the more important question is, what signals do cells communicate? And how does communication lead to specification? What's amazing is that across species, both vertebrate and invertebrate, there's a conserved system, a shared toolbox, if you will, that drives the specification and differentiation of cells. This system can be broken down into 7 key signaling paradigms that highlight the what and how of cell communication. These 7 conserved pathways are Wnt signaling. This can be broken down into canonical and non-canonical Wnt signaling. The next one is hedgehog signaling. These are super important embryonically. There are 3 different types. The most common by far is sonic hedgehog. We also have Indian and desert hedgehog. The next paradigm is transforming growth factor, TGF-beta. We also have notch signaling, receptor tyrosine kinases that I'll abbreviate RTK, retinoic acid signaling, and integrin signaling. I'll make separate short videos that walk through the basics of each of these pathways, but I just want to reemphasize the point that these are the pathways of embryonic development and cell differentiation. And not surprisingly, much of what we know about these pathways has come from cancer research. The effects of mutation or dysregulation of these foundational pathways and the aberrant growth that it drives in promoting cancer. But before we dive into specifics, there are a couple of patterns or trends worth noting. The first, which we introduced in our previous video, is the importance of concentration gradients, and the idea that the signaling molecules are seldom directional, although there are some cases where they are, but rather they simply diffuse from high to low. This has to do with the stochastic nature of movement and the fact that we can predict a net movement that searches out equilibrium. Now, I do think it's worth noting that according to Fick's law of diffusion, the rate of diffusion, if we were to draw this out in some sort of mathematical equation, is going to be directly proportional to the strength of the gradient. In other words, the steepness of the slope, right? This is the difference in concentration across a certain distance. With the steeper the gradient, the higher the rate of diffusion. It's also going to be directly proportional to the surface area. As an example, we see this in the lungs, where instead of 2 large balloons in my chest cavity, I've got millions of tiny balloons, alveoli, and this significantly increases the surface area so I can maximize diffusion of oxygen and carbon dioxide across the respiratory membrane. We're also going to be directly proportional to the solubility of the substance. And because we're talking about a biologic system, mostly this is going to be solubility in water. So things that are very soluble in water will travel better through water. But if I don't want something to travel very far, and we'll show this in a second in the case of sonic hedgehog, then I can alter its solubility. I can decrease its solubility, and that actually hinders its ability to diffuse. So those 3 things, strength of the gradient, surface area, solubility, these are all directly proportional to the rate of diffusion. There are a couple of things that are inversely proportional. So we can divide this equation by distance. Of course, that makes sense. The farther I have to go, the less likely I am to diffuse. And also size, specifically the molecular weight of the molecule that's diffusing, although this plays a little bit less of an effect. So in Fick's law, we actually see that this is the square root of the molecular weight. Now, why do we spend time talking about that? Well, This is important because it helps us see that there are several mechanisms by which diffusion of signaling molecules can be regulated. I can regulate the rate of diffusion based on size. And based on the relationship that we described, we can see that a decrease in size will lead to an increase in diffusion because these are inversely proportional. Also solubility, which is directly proportional, so a decrease in solubility will lead to a decrease in diffusion. And then the last thing that I want to mention really fast is the idea of gradient summation. Well, what do I mean by that? Let's say we have a gradient of particle x, high on the left, decreasing as we move to the right. But what if we have another gradient that maybe looks like this, and then another one, this is going to be small, maybe this one doesn't diffuse as far, we put right there. Where these gradients overlay, we could either see synergistic activity or antagonistic activity. We'll see this example with somite formation where we have several gradients involved. We have a notch gradient, we have competing interests with a retinoic acid gradient and an FGF gradient which cancel each other out. And this defines areas where we can make cuts in tissue to create somites. And so the idea of gradients and diffusion applies to all of the concepts that we're talking about simply because as these small molecule signal transducers are communicated, we have to keep in mind that they're all affected by these mechanisms. Now, the second concept I'd like to introduce is that of collinearity. Gene collinearity isn't very common, but it is essential for the proper patterning of the embryo during development. So how would we define it? Well, gene collinearity is simply the order of genes on a chromosome and how it correlates with the order of their expression in an organism's development. To give an example, perhaps the best example of this in humans is the Hox gene clusters. There are 4 such clusters in the human genome. We have one on chromosome 2, another on chromosome 7, another on 12, another on 17. To highlight one of these as an example, let's look at the HOXA cluster. This one's located on chromosome 7 and looks something like this. We'll draw a segment of the chromosome. Of course, we're going to read this from 5' to 3'. And let's look at the placement of the different Hox genes within this cluster, where each box represents a gene. Here we've got HOXA 13, followed by 11, 10, and 9, and then 7, 6, 5, 4, 3, 2, and 1. So we see the sequential order of these genes along the chromosome. Collinearity then describes that this correlates with their expression in an organism's development. So let's take a look at what that might look like. If we look at the developing embryo, in the case of Hox clusters, we want to look at the rostral caudal expression pattern. And what we can see is the segmentation of the embryo based on Hox gene expression, where Hox 1 and 2 are going to be expressed in the hindbrain and drive the differentiation of aspects of the hindbrain. Hox 3 is expressed in the lower hindbrain and drives the development of pharyngeal arches. This is going to contribute to the thymus and the thyroid. Hox 4 contributes to the development of the neck. Hox 5 is expressed in the lower cervical and upper thoracic regions and contributes to the development of respiratory and digestive structures in those locations. Hox 6 contributes to thoracic vertebrae. Hox 7 to muscles of the thoracic cavity. Hox 9 to lower thoracic and lumbar vertebrae. We also see Hox 9 expression important in limb development. Hox 10 is expressed in lumbar and sacral regions and also the hind limb. Hox 11 again in the limb, also the kidney, which is lower retroperitoneal. And then lastly, Hox 13 on the pelvic floor contributes to the development of the reproductive system. Again, this is just one of the 4 Hox clusters, and this gives us a sense, an idea of how gene collinearity works, with the order of genes on the chromosome correlating with their expression patterns in the organism's development. And again, while this is fairly rare, it plays a very important role in the embryonic patterning of the embryo. So to summarize, we introduced the idea of cell communication, that it can be direct and indirect, but we want to know what signals do these cells communicate and how does this communication lead to specification. We talked about 7 key evolutionarily conserved signal transduction pathways that we will describe later, each of them receiving their own little video. We talk again about the importance of concentration gradients and what might regulate the rate of diffusion. And lastly, the idea of gene collinearity, that in some cases, albeit rarely, the organization of the genes on the chromosome directs the expression that we see during the organism's development.